Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International A-Level Biology Revision topics, and this is topic 2B, Proteins and DNA. The first one is topic 2B1, which is talking about enzymes. Enzymes are biological catalysts, and their purpose is to control the rate at which chemical reactions occur within cells, outside cells, or within whole organisms. These enzymes work by speeding up the rate of chemical reactions without changing the conditions within the cytoplasm. You also need to know that enzymes are globular proteins. That is why they have specific sites, or the way the enzymes fold into the 3D structure allows them to have a specific shape. And as enzymes fold, they go through primary, secondary, tertiary, and mostly going to be quaternary structures. That allows them to have the 3D structure for the active sites in order for the substrate to bind into those active sites. Wherever enzyme-controlled reactions occur, factors like temperature, pH affect the efficiency at which these enzymes perform specific tasks. So if these factors affect the intramolecular bonds within the proteins, like ionic bonds, hydrogen bonds, this can be responsible for changing the shape of the enzyme and therefore affecting the active site of the enzyme, causing the enzyme to be denatured. Next, we go to naming enzymes. Enzymes can be intracellular or they can be extracellular. Intracellular enzymes catalyze reactions inside cells. Examples here we have DNA polymerase, DNA ligase, we have helicase, and so on. But for extracellular enzymes, these are mainly involved in reactions outside the cell, like those in the digestive system. These 4 points are related to naming enzymes, so usually the enzymes we see have the word "ase." This is added to the end of the substrate. For example, we have lipase. This is for digesting lipids. We have urease that works on urea, and so on. Sometimes enzymes can be named by adding an indication of what it does. For example, creatine kinase. Sometimes an enzyme, there should be an enzyme, can be given a longer name describing the type of reaction that is being catalyzed. For example, creatine phosphotransferase. Also, some enzymes can be given a classification number. For example, you have this in your textbook. So sometimes if you want to buy specific enzymes, you use a classification number instead of the actual name. And some enzymes like trypsin and pepsin, hearing the names trypsin and pepsin, you cannot know the role or even the type of reaction that these enzymes catalyze. So for some enzymes, their names do not give any hint to the roles that they perform in the human body. So for topic 2B2, we go to how enzymes work. We need to know that enzymes participate, of course, in chemical reactions, but for chemical reactions to occur, activation energy has to be attained. Activation energy is the minimum amount of energy that is required to break the chemical bonds holding the reacting substances together. In this case, we are talking about reactants, or you would say in the substrate. So reactants, or biologically, we can say substrate. So in this case, enzymes work by lowering the activation energy needed for specific reactions to take place in order to form an enzyme-substrate complex or in order to form the products. So for example, we have this graph showing us this is the pathway for the uncatalyzed reaction or a reaction without an enzyme. You can see it goes towards until where the products are. But when a catalyst is used, or in this case when an enzyme is used, we can see the pathway is going to be this way until we see the products. So basically, it's only this area that changes. The reactants and the products are still at the same point for both pathways. The difference is we can see for pathway 1, where there is no enzyme, this is going to be a higher gap, and this gap represents the activation energy, which we can see here. So uncatalyzed activation energy is going to be higher compared to the activation energy that is required when an enzyme is used. So we can see that using an enzyme causes the reaction to proceed, requiring lower activation energy, so temperature used and so on is going to be lower than would have been used if an enzyme is not used. So let's go to the hypothesis of enzyme activity. We know there is one that is called a lock and key model, and in this model, the enzyme and the substrate are going to be complementary, meaning the active sites of the enzyme are a perfect fit for the substrate that is going to be worked on. You can see the shape is exactly the same. The substrate is going to fit in because the shape is complementary to that of the active sites of the enzyme. So in this case, the substrate fits into the enzyme and an enzyme-substrate complex is going to be formed. A reaction takes place within the active sites, products are formed, and then the products are going to leave the active site of the enzyme. In this process, the enzyme is not changed. In the induced fit hypothesis, we can see that they are not necessarily complementary. I'm talking about the active site as well as the substrate. they may not be exactly complementary, but as the substrate comes and fits into the enzyme changes or is going to readjust itself so that the substrate can fit within the active sites, and then a reaction takes place, and after the products are going to move away or they're going to be removed from the active site of the enzyme, and then the shape will be restored. This is called an induced fit hypothesis because there is change in the shape of the active site to accommodate the substrate that has come in order for a reaction to be catalyzed. Moving on. When we go to factors affecting enzyme activity, like I already said, I talked about pH already, as well as the temperature. So we already know that enzymes speed up reactions. Only very little amounts of these enzymes have to be used in order for reactions to be catalyzed or for the substrate to be converted into products. And during enzyme activity, there is a term called molecular activity of an enzyme, which is going to be the measure of the number of substrate molecules transferred per minute by a single enzyme. So usually enzyme catalyzed reactions, they have a higher molecular activity or a higher turnover number because fewer enzymes are required for many substrates to be converted into products. We also need to know that enzymes have active sites, and these active sites are the ones onto which the substrate is going to bind, like I already showed you before, so that a reaction can take place. Some of these active sites are so specific to catalyze only one particular reaction. So changes in conditions, like we already say, like temperature, pH, can affect the shape of the active site, and when they do, it means the enzyme and the substrate can no longer be complementary, therefore an enzyme-substrate complex cannot be formed. For these enzyme-controlled reactions, factors like enzyme concentration, substrate concentration, temperature, and pH have a great role they play in the amount of substrate that can be converted into product or in the rate of enzyme control reaction. So here we're looking at some of the factors. We're beginning with substrate concentration. Of course, the higher the substrate concentration, the higher the rate of enzyme-controlled reaction until the enzymes are fully saturated. So we can see here on the horizontal axis, we see substrate concentration is increasing, and we see as substrate concentration increases, the rate of reaction increases until a specific point when it kind of levels off. The point at which it levels off is when all the sites of the enzyme are fully occupied, and no matter how much the substrate concentration increases, since the concentration of the enzymes is constant, we have no more active sites that can be occupied, and therefore the rate is going to be affected by the number of active sites. So I would say the active sites are going to be limiting the rate of reaction when this slows down in order to level off. The next one is about temperature. We know enzymes are temperature sensitive, so in this case we see the higher the temperature, the higher the rate of enzyme controlled reaction until this specific point, which we call the optimum temperature. After the optimum temperature, we see that no matter how much we increase, the rate of enzyme controlled reaction decreases. Then this is because the active sites have been affected, or higher temperature causes the breaking of the hydrogen bonds or other bonds holding the 3D structure of the enzyme together. So this causes the active sites to be lost, and when they are lost, an enzyme substrate complex can no longer form, and therefore the rate at which substrates are converted into products is going to decrease, so the rate of reaction slows down. Also, when we look at how temperature affects the rate of reaction, we'll look at the factor which we call the Q10. The Q10 is the rate of reaction at a specific temperature plus 10 degrees divided by the rate of reaction at that particular temperature. For example, if the particular temperature we're calling X here is 10 degrees, so here the numerator is going to be rate of reaction at 20 divided by rate of reaction at 10, and that will give us the Q10. So the effect of temperature on the rate of any reaction can be expressed using that coefficient we call the Q10. Like I wrote here, for any reaction between 0 to 40 degrees, the Q10 is going to be equal to 2 because the rate is going to double for every 10 degree rise in temperature. And this can be observed using this graph here. In the exams, they can tell you to calculate the Q10 for some reactions, so please, you have to be able to read from the graph. If we have 10 degrees here, this is going to be the rate at 10 degrees, that will be the rate. Then if we have maybe 10 plus 10 is going to be 20, so at 20 degrees, if this is 20 degrees, you're going to come to the graph and then to the other side, that is going to be the rate at 20. So to find the Q10, it will be the rate at 20 divided by the rate at 10, and that is going to give us the Q10. Moving on, this pH. We also know different enzymes work within specific pH ranges, so this is not similar to the one we saw with temperature, where we see the optimum temperature is going to be almost the same for most of the enzymes within the human body, but for pH it's a different story. So here we see change in pH affects the formation of hydrogen bonds, of course, and disulfide bonds within an enzyme, And you remember hydrogen bonds, disulfide bonds hold most of the 3D structures of that protein, which is actually the enzyme. And if changes in pH affect the bonds, it means they're going to affect the 3D structure and they're going to alter the active sites. And that means the enzyme is going to be denatured because an enzyme substrate complex can no longer be formed. Similar to what I've said already, different enzymes have different pH values or pH ranges within which they can work optimally. The examples here we have cholinesterase. We can see it's going to have a maximum activity at about this point. You can see the pH is between 8 and 9. We also have here trypsin. Of course, this is going to be the optimum, again between 8 and 9. But we have pepsin. The optimum is going to be between 3 and 4. So you can see all these enzymes have different optimum pH ranges for when the rate of reaction is going to be the maximum. To topic 2b3, the structure of DNA and RNA. Mononucleotides are very useful in the body in the formation of many molecules like ATP, adenosine triphosphate, which has 3 phosphate groups. And then there is a pentose sugar, which is a 5-carbon sugar. And then we have a nitrogen base, like we're going to see something here. They're also useful in the formation of DNA as well as RNA, DNA being deoxyribonucleic acid and RNA being ribonucleic acid. Each nucleotide or each mononucleotide has 3 parts. There is a 5-carbon pentose sugar, which is similar to what we have here. And then there is a nitrogen base, this one here. Nitrogen bases can be purines as well as pyrimidines, like the purines include adenine and guanine. The pyrimidines include cytosine, thymine, and uracil, although this one here is found in RNA, not DNA. So these two are exchanged for each other in RNA and DNA. In DNA, we have thymine, and in RNA, we have uracil. We also have a phosphate group, which is going to be this in a mononucleotide. In RNA, the pentose sugar is going to be ribose, like we see here, and in DNA, that pentose sugar is going to be deoxyribose. The difference between the two is there is no oxygen here, but we have OH, so there is OH here and only a hydrogen here. That is the difference between ribose and deoxyribose. This is found in DNA and this is found in RNA. When we go to polynucleotides, these are formed when many mononucleotides are linked together. So these are nucleic acids, which are formed within cells. They are polymers consisting of many mononucleotides, like the ones we saw on the previous page, held together by phosphodiester bonds. The chromosomes in the nucleus of eukaryotic cells, like, of course, our own, will store the genetic material in the cord. Parts of this cord are used as a template to make complementary strand. Of course, we'll talk about this later. Parts of the cord are used to make messenger RNA, which is later going to be user translated in order to produce proteins that are used as building blocks for cells, as well as controlling specific actions within our bodies. But we have this polynucleotide. We see the phosphodiester bond. We can see that's one example. And then we can see the many mononucleotides like this one here being added to the group, which is that, and then the other, and then the other. So together we can form the polynucleotide as many mononucleotides are linked together. To compare RNA and DNA molecules consist of 2 polynucleotides. Like you can see, it's a double strand. There is one strand and the next strand. The sugars and the phosphate form the backbone. We're talking about this being the phosphate and that being the sugar. So we see this is going to be the backbone and the nitrogen base are going to be the links in between. So the sugar and the phosphate form the backbone of the molecule. Pointing inwards from the 2 sugar or phosphate backbones are the bases, which are paired in the specific ways. For example, in a DNA molecule, the adenine pairs with thymine, while the cytosine pairs with guanine. We do not have uracil in DNA, only the other 4. A purine base always pairs with a pyrimidine. Yeah, for example, you remember we talked about the purines, adenine and guanine, and then the pyrimidines with the cytosine, uracil, and thymine. But we know in DNA we do not have uracil, so DNA adenine will pair with thymine, and cytosine will pair with guanine, as you can see here. Thymine with adenine and cytosine with guanine on either strand. This results in the famous double helix, like you can see here. It's a double helix, 2 strands joined together by hydrogen bonds in between them. The 2 strands of the DNA double helix are held together by hydrogen bonds between the complementary bases. These are hydrogen bonds holding together the nitrogen bases on either strand. For RNA, the molecules form a single polynucleotide, meaning it's going to be 1 strand. So the RNA molecules have base uracil instead of thymine. So these can either fold into complex shapes, I'm talking about the RNAs, or they can be held in place by hydrogen bonds, or they can remain as long thread-like molecules. Moving on to topic 2b4, how DNA works. There are two main ideas about how replication happens. One is called the conservative replication, and the other is the semi-conservative replication. In the conservative replication model, the original double helix remains intact, and in some way it instructs the formation of a new identical double helix made entirely of new genetic material. So this means the new double helix does not contain any genetic material from the original double helix. However, in the semi-conservative replication model, we assume that the DNA is going to unzip and new nucleotides are going to align along each strand. Each new double helix is going to contain one strand from the original DNA and one strand made up of the new material. This is the Watson and Crick hypothesis. So to test which hypothesis occurred during replication, E. coli was grown in media containing only heavier nitrogen, or you'll call nitrogen, with isotope nitrogen-15 after several generations of culture, or the bacteria contained only that specific nitrogen. So the bacteria was then cultured in the lighter nitrogen-14, and the density of the DNA produced was measured after one replication cycle. After one replication cycle, our DNA had the same density halfway through 14 as well as 15. So they concluded that replication occurred through the semi-conservative model, where the DNA strand unwinds and zips, and then nucleotides are going to attach in order for the new double helices to be formed, each containing a strand from the original DNA. So here, this is a demonstration of the two models. This is the conservative, where we expect the original strand, this is the original, to somehow instruct the formation of the new strand. This is going to be the new strand, purely 100% lighter nitrogen, and the other is going to be the heavier one. If this was heavy, then this is going to be heavy, and that is going to be lighter, like we can see here. This is nitrogen-15, which is heavy. This is going to be 15, meaning that remained exactly as it was. It did not unzip. And then we're going to see nitrogen-14. This is on the lighter side. If this is the intermediate for 2, here we can see the new strand is going to be 100% 14, and the other is going to be 100% nitrogen-15. There is no halfway through both of them. That is the conservative model. When we go to the semi-conservative model, the DNA is going to unzip, and then each is going to form a new individual double helix as new nucleotides attach onto the original strand. This is the lighter nitrogen, and then we can see this is sample 1 only has heavier nitrogen, so we can see this is heavy DNA. So after one replication cycle, remember this is in nitrogen-14, which is going to be lighter. We see it's going to be 100% intermediate, and that shows us that each of these two strands split apart in order for newer nucleotides to attach onto each, so that everything is 100% intermediate DNA, or the hybrid strand, which we see here. In the next replication cycle, we see we have 50% nitrogen-14, and we see 50% a hybrid or mixture of the two. If I can show you here, this means these strands broke apart, producing this one here and that one there, and each paired with a new strand made up of new genetic material to produce 100% of the hybrid. In the next cycle, this split into be that split to be this, that split to be that, and that split to be this, and each paired with a new nitrogen-14. And the two here are going to be 50% of the hybrid, which we see here. Looking at the real DNA replication, we can see this is the double helix. So we see there is going to be unzipping. So this is going to be carried out by DNA helicase. This is an enzyme that carries out the breaking of the hydrogen bonds holding the two strands together. Once these hydrogen bonds are broken, then DNA polymerase is going to cause the lining up of the new nucleotides to produce the new strand. And then DNA ligase is going to be able to act as genetic glue in the formation of the phosphodiester bond between the specific nucleotides that are being paired up to create a new strand. Going to topic 2b5, the genetic code. So in the DNA double helix, we know the backbone is going to be exactly the same, made up of a phosphate and a sugar, but the bases are the ones that vary. We know that there are 20 naturally occurring amino acids, and these have to be joined together in complex combinations in order to produce infinite varieties of proteins. We know there is going to be transcription and then translation occurs on the ribosomes as the amino acids are assembled in order to produce the protein. We know we have 4 bases and we have 20 amino acids. Yet each codon has only 3 bases. The allowance is only for 3 bases. So it means we have 4 bases for the first position among the codon. This is representing the codon. Then there is 4 other bases that can be positioned for the second and 4 others for the third, which gives us 64 possible combinations. And this is going to be more than enough to cater for the 20 amino acids that must be coded for. We have genes, which are a sequence of bases of a DNA molecule which code for a specific amino acid that is supposed to be part of a polypeptide chain. Each of the 3 bases along a strand of DNA is going to code for a very specific amino acid. Of course, we know the codon, we've already talked about it, 3 bases. But actually, some of them signal the beginning or the end of a particular amino acid sequence, meaning there is a start codon as well as the stop codon. So some of these triplets could be coding for a start codon and others could be coding for a stop codon. So sequence of 3 bases on a DNA or RNA, this is what we call a codon. In the human DNA, 98% of this is non-coding, meaning these are DNA sequences that are involved in regulating protein coding sequences, effectively turning genes on and off. So these 98%, some of them scientists do not know what they exactly do, but we know they are involved or could be involved in the turning or switching on and off of genes. Only 2% of the human DNA is what codes for specific proteins. So some of these code for a particular amino acid, while others, like I already said, code for the beginning or ending of a particular amino acid sequence, which is the start codon as well as the stop codon. The properties of the genetic code. The genetic code is only a triplet, meaning 3 bases, or it is read as 3 bases at a time. It is also non-overlapping and is going to be degenerate. Now, these 2 qualities are very important most of the 6-mark questions. When they say non-overlapping, it means you read them 3 at a time, and a base that appeared in the previous codon does not pass on to the next codon. For example, if I have AGG, CTC, something like that, and maybe AGC, it means I have to read them as 3, 3, and 3. I cannot do 3 and then come and read the next 3 and then the next 3. That will be overlapping and that is going to be wrong. So this is wrong, that is wrong. We read them 3 at a time, and a base from the previous codon does not show up in the next codon. It means 3 bases at a time. That is to be non-overlapping. They are degenerate because they code for more than necessary. For example, again, if I have AGG and CTC, this means that mainly the first 2 bases are the ones that are going to be representative of a specific amino acid that is coded for. So it's like there is more information than actually required within the genetic code. That means degeneracy. Topic 2b6, DNA and protein synthesis. So here we begin with RNA. Of course, we're going to look at protein synthesis, so RNA is very important. RNA contains a different sugar. Remember, this is going to be ribose and not deoxyribose like we saw in DNA. And it has a different base, which is going to be uracil instead of thymine. I already talked about this. RNA has some specific functions, like it carries out instructions of a polypeptide from the DNA in the nucleus to the ribosomes where proteins are going to be made, meaning RNA takes part in transcription as well as translation. It means the information in the DNA is going to be converted into messenger RNA, and then this is going to leave the nucleus and take the information with it to the site where protein synthesis occurs. Also, RNA picks up specific amino acids from the protoplasm and carries them to the surface of the ribosome. This type of RNA is the transfer RNA. So we have seen the messenger RNA, now the transfer RNA. And then RNA makes up the bulk of ribosomes. This is what we call ribosomal RNA, meaning it makes up the ribosomes. Actually, ribosomes are ribosomal RNA as well as protein mixed together. Here, I say it again, we have messenger RNA, we have transfer RNA, and ribosomal RNA, as we're going to see each of them in detail. Beginning with messenger RNA. Messenger RNA is formed in the nucleus during a process of transcription. To transcribe is like making a copy of something. So information from DNA is copied onto a single strand of RNA, which we call messenger RNA. We can see there is unzipping, the part or section of DNA unzips, and then RNA polymerase attaches nucleotides onto that place. This is RNA polymerase. We see the nucleotides being added, phosphodiester bonds are formed, and then what results is going to be a messenger RNA. This all occurs on the antisense strand and not on the sense strand. So the messenger RNA carries information that codes for a specific polypeptide. Transcription occurs at a start codon and ends at a stop codon. We can see this is the process of transcription that is being described here, making a copy of messenger RNA. The formed messenger RNA, there should be no space here, the formed messenger RNA separates from DNA and moves to the surface of the ribosome, basically out of the nucleus to the cytoplasm, to the surface of the ribosomes, where protein synthesis is going to take place. The next type of RNA is transfer RNA. This is involved in the formation of the polypeptide. So this transfer RNA is going to be found in the cytoplasm, and it has a specific shape that is different, as you can see here. One part of the transfer RNA has a sequence of 3 bases that match the genetic code. We call this the anticodon. This anticodon is what is going to bind on the codon of messenger RNA. If this is messenger RNA that comes onto the ribosome, these 3 bases here are going to be the codon. So the anticodon and the codon have to be complementary. So if we have adenine here, this has to be uracil, because you remember we do not have thymine in RNA, so uracil is going to be the one that binds with adenine. In cytosine, this has to be guanine, and if this is guanine, this has to be cytosine as well. I'm just using this as an example, so they have to be complementary in order for them to bind to each other as the new amino acid is added to the chain. This is going to be the site of the amino acid binding, so the amino acid binds there, and then it's going to be added onto the growing polypeptide. And so there is going to be a lot of amino acids that can be carried through the ribosome where translation is going to occur. This is the process of translation, meaning addition of many amino acids to the growing polypeptide in order to produce the protein. Moving on to ribosomes. Ribosomes are made up of a combination of ribosomal RNA as well as proteins. I talked about this a little bit before. They consist of a large unit as well as a small unit. I would say translation part of protein synthesis occurs. So the messenger RNA is going to bind into the smaller and upper units of ribosomal RNA, or the ribosomes, in order for transfer RNA to come with its anticodon, bind onto the codon of the messenger RNA in order for the amino acids to be added to the growing polypeptide. So the ribosomes surround and bind to parts of the messenger RNA that are being actively translated and then move along to the next codon. Their job is to hold together messenger RNA and transfer RNA and act as enzymes controlling the process of protein synthesis. During protein synthesis, the genetic code or DNA is going to be transcribed or copied onto messenger RNA during the process of transcription. So, after transcription, with the help of RNA polymerase, messenger RNA is going to be formed, and these will move out of the nucleus into the cytoplasm and become attached to the ribosome. The ribosome is the site for translation, or where the amino acids are added to the growing polypeptide. So the molecules of transfer RNA, these carry individual amino acids to the surface of the ribosomes. And again, different transfer RNAs have different anticodons that are complementary to the codon on the messenger RNA, and only complementary transfer RNAs will bind because they contain the amino acid that has to be added at that specific position within the growing polypeptide chain. So the transfer RNA anticodon lines up beside a complementary codon on the messenger RNA, held in place by the hydrogen bonds while enzymes link the amino acids together. Remember, we have the transfer RNA and messenger RNA. So we have the codon anticodon. They are going to form hydrogen bonds between them as the amino acid is added to the growing chain. The transfer RNA then breaks away and returns to the cytoplasm to pick up another amino acid. Then the ribosome moves along the molecule of messenger RNA until it reaches the stop codon at the end of the polypeptide. So when a stop codon is reached, then that means there is no more amino acids to be added, so the polypeptide chain is complete. There is a chance that this message may be read again and again, sometimes to make more proteins of the same kind. So these are the details of the process of protein synthesis. We can see The first step is transcription. Here the DNA information is going to be copied onto messenger RNA. There is a sense strand and the antisense strand. The antisense strand or the template strand is the one that is going to be copied or used in the formation of messenger RNA. RNA polymerase is going to help during the formation of the messenger RNA. The formed messenger RNA is going to leave the place of DNA, it's going to leave the DNA molecule, and then it's going to get out of the nucleus and go to the ribosome. So in step 2, we see messenger RNA carries the information to the ribosome. The completed messenger RNA strand is going to leave the nucleus to the ribosome in the cytoplasm because that is where translation is going to occur. In step 3, where we have amino acid activation, we see transfer RNA is going to carry amino acids to the ribosomes. As we already know, each transfer RNA is going to have a different anticodon that is complementary to the codons we see here on the messenger RNA. This is our messenger RNA. It has specific codons, and this incoming transfer RNA has an anticodon that is complementary. For example, guanine has to pair with cytosine, uracil with adenine, and guanine with cytosine. So we can see this anticodon and this codon here are going to be complementary, so that specific amino acid is going to be at the right position within that specific polypeptide that is being formed. So we see this is the growing chain of the amino acids forming the polypeptide, as more and more are going to be added within the chain in order to produce the protein, and that process is what we call translation. Now in the end, that completed polypeptide is going to fold, or it's going to be modified in order to produce a functional protein. So in this case, it could form a specific shape. If it's going to be maybe an enzyme, specific molecules could be added to it if it's going to be a specific protein. So basically, modification of this protein or polypeptide is going to be carried out in order for it to fulfill its function appropriately. So this brings us to the end of this topic. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.